Hello students, I am Dr. JAYASHREE, Assistant Professor, Department of Biotechnology, School of Bio and Chemical Engineering, Sathyabama Institute of Science and Technology. Today we are going to discuss about G-protein coupled receptor. Before going to deep about G-protein coupled receptor, we have to know the function of receptor. Receptor help to pass the information, extracellular information into intracellular response. Receptor can be different types. It can be classified into two categories based on the where it is present. It may be classified into cell membrane receptor and intracellular receptor. Cell membrane receptor found in plasma membrane, whereas the intracellular receptor present in nucleus and cytoplasm. So coming to the introduction about G-protein coupled receptor, also called as GPCR, or the largest and most diverse group of membrane receptor in eukaryotic plants, microorganisms and invertebrates. GPCR are the most major class of cell membrane receptor that are found associated with the group of G protein consisting of three subunits termed as alpha, beta and gamma. GPCR also represent the largest, more efficacious class of therapeutic target for disease including cardiovascular disease, cancer and asthma. GPCR are the integral membrane protein that are used to convert extracellular signal into intracellular response. It contains 7 membrane spanning helical connected by 3 short extracellular loop and intracellular loop. So they are also known as 7 transmembrane receptor. 2 highly conserved cysteine residue will help to stabilize the receptor structure. G-protein coupled receptor, also called as 7-pass transmembrane domain receptor, 7-TM receptor, heptahelical receptor, serpentine receptor due to its snake-like structure, and also called as G-protein linked receptor. The structure of a G-protein coupled receptor, this image clearly show you the 7 transmembrane present in between the plasma membrane, and N-terminal end and C-terminal end will be present. In extracellular place, the ligand binding site is shown clearly. In cytoplasmic place or intracellular surface shows the G-protein binding site. There are two principal signaling transduction pathway involved in G-protein coupled receptor. One is CAM pathway, another one is phosphatidyl inositol signaling pathway. The G protein are specialized protein which able to bind with nucleotides like guanosine triphosphate and guanosine diphosphate called as GTP and GTP. It is the G protein is in active form, it will be bounded with the GTP. In inactive form, it will be bounded with GTP. The G protein that are associated with 3 different subunits, I already mentioned alpha, beta and gamma subunits. In this, alpha and gamma are attached to the plasma membrane by lipid anchor. The ligand binding to the GPCR receptor causes change in the receptor conformation that it bind turn and to activate the G protein. This image can clearly explain the activation form of G protein. The signal molecule, it may be a ligand, it may be a drug molecule, it may be a hormone. It will come and bind with the signal receptor and it will activate the receptor. The conformational change lead to the give the instruction to the intracellular process. So it will activate the inactive form of G protein to active form of G protein. The inactive form will be alpha, beta and gamma will be combined together. It is called trimer. So GDB will be founded. If it is the alpha subunit binded with GTP, it is called as inactive form of G protein. If it is activated by the instruction by the signal molecule, the GDP will be transferred to GTP. So this leads the conformational change in the G protein and it will come to active form. In its active form, the alpha subunit will be bounded with GTP and then beta and gamma will be separated. Each of the subunit have their functional role in the cellular signaling process. The active form of the G protein that is released from the surface of the receptor dissociating into alpha and beta and gamma subunit. Both subunit will be activated their specific effector causing the release of secondary messenger. These secondary messenger are recognized by the protein kinase leading to the activation and trigger the signals cascade toward the cellular response. Further, affect intracellular signaling protein or target function protein directly depend on the subunit type. there are different type of alpha subunits are there. These I have mentioned GS, GI and GQ. These are the importance alpha subunit which will be for the importance secondary signaling process. And beta and gamma. This beta and gamma directly go and bind to the ion channel which will be present in the plasma membrane. Coming to GS, GI, GQ. GS will be activate the adrenergic cyclase activity. GS will inhibit the adrenergic cyclase activity. So one receptor can stimulate or inhibit the process. When coming to GQ, it will activate the PLCB, protein lipase C beta enzyme. This image, can you explain the full process of G protein coupled receptor activation under the subunits roles of subunit? So here first the ligand is binding with the GPCR. The GPCR will be inactive form will be activated. When it is activated, GTP which bound with alpha subunit bounded with GTP, and then beta and gamma will be separated. This beta and gamma directly go and bind with the ion channels which are present in the membrane, plasma membrane, and open the ion channels and transfer the calcium and potassium inside and outside of the cell. So when coming to the G alpha subunit, it has different alpha subunit. First important one is GS, second one is GI and GQ. Here GS will activate the adrenergic cyclase enzyme. So it will activate the adrenergic cyclase enzyme. Due to this, the CAMP production will be increased inside the cell. When coming to the GI, it will reduce or it will stop the adrenergic cyclase enzyme. So it will control the cyclic AMP production in a cell. When coming to GQ, it will go and activate the phospholipase C enzyme. So it will lead the production of calcium ions in the cell. So this CAMP and calcium ions are placed as secondary messengers. These secondary messengers are giving a further instruction to the intracellular process. These are all the reference images and papers I have taken for your reference. Thank you all.